During anaphase of the first round of meiotic division, homologous chromosomes separate and migrate to opposite poles. During anaphase of the second round of meiosis, and this applies to mitosis as well, sister chromatids separate and move to opposite poles. Meiosis is responsible for producing 4 haploid cells from one original diploid parent cell. Failure of chromosomes to separate from one another during either round of meiosis results in non-disjunction. This can occur in meiosis or mitosis in either sex. Normal chromosome segregation begins with a diploid cell at the start of meiosis. We will focus on one pair of homologous chromosomes as they segregate. This pair can represent either autosomes or sex chromosomes. Each chromosome consists of 2 sister chromatids joined at their centromere. During the first meiotic division, homologous chromosomes separate, resulting in 2 cells, each containing one pair of sister chromatids. During the second meiotic division, the centromeres split and the 2 sister chromatids segregate to different cells. The end result is 4 haploid cells, each with one sister chromatid, now called a chromosome. In animals, these 4 haploid cells are called gametes. When they fuse with a gamete from the opposite sex, normal diploid cells are produced. Let's observe what happens when non-disjunction occurs during meiosis I. If the homologous chromosomes fail to separate, then one cell is produced with both homologs, and the other cell has no copies of this chromosome. When the cell divides during meiosis II, the centromeres of both sister chromatids split and they segregate to each of the 2 progeny cells. These 2 cells are unusual in that they contain 2 copies of this chromosome instead of 1. In the other 2 cells produced, there are no copies of this chromosome, instead of the usual 1. Upon fusion with a normal haploid gamete, all of the cells produced are abnormal for this chromosome. Half of the cells will have 3 copies of this chromosome instead of the normal 2. These cells are said to be trisomic for this chromosome, and the condition is called trisomy. The other half of the cells will have only 1 copy of this chromosome. These cells are said to be monosomic for this chromosome, and the condition is called monosomy. Let's observe what happens when non-disjunction occurs during the second meiotic division. Homologous chromosomes separate normally during meiosis I, producing 2 cells, each having a pair of sister chromatids. If the sister chromatids in one of the cells fail to separate during meiosis II, then 2 abnormal cells are produced, one having 2 copies of this chromosome and the other having none. Normal chromosome segregation occurs in the other cell, giving rise to 2 normal haploid cells. When these cells fuse with a normal haploid gamete, 2 of the cells produced have the normal set of chromosomes and produce diploid cells. The other 2 cells formed are abnormal, one having 3 copies of this chromosome, trisomic, and one having only 1 copy, monosomic. In sum, non-disjunction produces an abnormal number of chromosomes in a diploid cell. It may occur either during meiosis I or meiosis II. If it occurs in meiosis I, all 4 cells produced have an abnormal number of this chromosome. If it occurs in meiosis II, 2 cells have the normal number of this chromosome and 2 cells have an abnormal number. Non-disjunction of the sex chromosomes is responsible for a variety of human disorders, such as Turner syndrome, a female with only 1 X, and Klinefelter syndrome, a male with 2 X's and 1 Y. Non-disjunction of the autosomes is responsible for Down syndrome, trisomy 21.